the rest of the time we'll introduce the gram-negative cocci, the Neisseria, and they are diplococci like this. This is not a real picture, it's an art picture. So diplococci like this, the cell look like the bean, right? The coffee bean. And they are always in pairs. And after staining, it's gram-negative. And we'll learn two different species. One is the Neisseria gonorrhoeae, or for short, we call them gonococci, the general name. Neisseria meningitis, as we call them meningococci, sorry. And these two species, gonococci and meningococci, are closely related, and over 70% of their DNA are the same, homologous. So this is the difference between the two species. Okay. Capsule, meningococci produce a capsule, but gonococci does not. Plasmid, gonococci carry a plasmid. Meningococci no plasmid. And also the clinical presentation. The entry to the body is different. Meningococci get into our body through the upper respiratory tract, but they can cause systemic infection, cause into the bloodstream, and also get into the brain. While the gonococci causes a genital infection, so it's a sex-transmitted disease. Let's talk about this in detail. First, meningococci. This is a gram stain, you can see. And in this specimen, you will not only see the bacteria, but you will also see cells like this. This is the nucleus of neutrophils, okay? The typical nucleus of neutrophils. And in this neutrophil, the whole area, this is a cell. And you can see some intracellular bacteria. So this is inside of the neutrophil cell. And it's gram-negative, diplococci, okay? And usually you can find intracellular bacteria, but sometimes you can also find some extracellular bacteria. And they are not motile. To culture this bacteria, we need to use the presence of oxygen because they are aerobic. They produce oxidase. They grow on chocolate agar. They also produce the autolytic enzymes that results in the rapid swelling and lysis. There's another thing I want to mention for this bacteria is that they prefer warm temperature. Cold temperature will kill them. And they don't like low temperature, they don't like dry condition, they like warm, moisture condition. So once you collect the specimen, you want to keep the specimen warm and transport the specimen to the lab. And when you culture the bacteria, you need to even pre-warm the medium, like the chocolate agar. We keep the chocolate agar in fridge usually. And before you use it, you want to warm it up in the incubator, then stick the bacteria on a warm plate. If you stick the bacteria on a cold plate, the bacteria will die. So they prefer the warm temperature. Antigens of this bacteria, the most important one is the capsular antigen. We mentioned that in that table that meningococci can produce a capsule. At least 13 serogroups of meningococci have been identified right now. And disease, the pathogenesis of meningococci. Humans are the only natural host for meningococci. to cause disease. And they enter our body through the upper respiratory tract, the nasal pharynx. The organism may reach the bloodstream, producing bacteremia. We know that that's the presence of bacteria in the bloodstream. The symptoms may like those of the upper respiratory tract infection, a common cold, but it can cause more severe disease like meningitis. That's the most common complication of meningococci infection. It usually begins suddenly with intense headache, vomiting, and progresses to coma within a few hours. So the onset of disease is sudden and the progress is quick. And also during the meningococcosemia, when they get into the blood, they can cause a lot of thrombosis in the small blood vessels, because the blood clot in small blood vessels in many organs, which might lead to serious problem. Okay, laboratory test. We collect the blood. They can form blood bacteremia or spinal fluid, and we make a smear and check, and you can see the typical Neisseria cell within the neutrophils. And sometimes you can also see the extracellular bacteria. And to culture the bacteria, we can use this chocolate agar. This is not a typical chocolate agar. It's like blood agar. But so use chocolate agar to culture this bacteria. Okay. To treat meningococci infection, penicillin G is the first choice. Previously, some students asked me a question: why penicillin G? Because penicillin G inhibits the cell wall synthesis in gram-positive bacteria, but this is a gram-negative bacteria. The answer is probably right now we don't understand the mechanism of penicillin G completely. So it's a broad spectrum antibiotic. It can inhibit most of gram-positive bacteria, but it also works on gram-negative bacteria, some gram-negative bacteria. So there is a new paper published several years ago starting the new mechanism of penicillin G, and probably there's something we don't know right now. So they might function in a different way to kill the bacteria. And you can also try other antibiotics such as chloramphenicol. It's a common antibiotic, but this antibiotic might have some side effects. So if possible, you use penicillin G. The antigenic structure of this bacteria, it produces penicillin. And we know that penicillin is the major factor for attachment or the adherence attached to the host cell, so that it is also resistant to phagocytosis. Another antigen you want to remember is the LOS. This sounds familiar, right? Lipo-oligosaccharide. We learn another term, LPS, right? LPS, that's a lipopolysaccharide. So what's the difference between oligo and poly? Poly means more, right? While oligo means several. So usually here, the oligosaccharide means it contains fewer saccharide molecules in it, while polysaccharide means it contains more saccharide molecules in it. And LOS involves in the toxigenicity of gonococcal infection. So that's a major virulence factor. the disease. Humans are the only natural host, okay? But in human population, some people does not produce disease. After infection, they will become asystemic carrier, and that is the major reservoir of this pathogen. And this reservoir, okay, mostly happen in female. So some female, after they get infected, they don't develop disease, but they become a carrier. Usually in male, it will turn out with active infections such as urethritis with yellow creamy pus and painful urination. So some women might become the asystemic carrier. Okay, but in a lot of female, it can also cause disease, such as the vaginitis and other inflammations, and it's a common cause of sterility. You understand the word of sterility? Right, the female cannot get pregnant. Okay, that's sterility. Okay, a rare type of infection is in a newborn baby. It's the gonococcal ophthalmia. So ophthalmia is the eye infection by gonococci. So that's when the baby is born to pass through the birth canal. If the mom is a carrier, the bacteria can get into the eye to cause infection. Without treatment, this little baby might suffer from blindness. But right now, the strategy to prevent such infection, we don't actually screen the mom to give the mom a test to see whether it's positive or not. We just treat the newborn baby with the eye drop, the antibiotic eye drop. No matter the mom is positive or not, we give every baby some eye drop, some antibiotic to prevent such infection. So it's more effective and it's more economic to save money because several drops of antibiotic does not cost a lot. I mean, without treatment, it might cause blindness. Probably not. It's not a systemic usage. It's just several drops into the eye. So, who's talking there? Okay. For laboratory test, we collect the pus and secretions from the urethra, cervix, and those material, and make a smear and check, and we will find many diplococci, gram-negative, and within neutrophils, a lot of neutrophils. And culture, again, we need to culture the specimen immediately. They can also autolyse themselves. And you can also run other tests like a DNA test, nucleic acid test, or serological test. Keep that in mind. You don't have to know the detail. For treatment, again, penicillin G is the first line medicine. And if the isolated is resistant to penicillin, actually this bacteria is developing more and more resistance. That's the current problem. I used to work not in the lab working on this. The next door lab is working on this. They are dealing with the antibiotic resistance. If it's resistant to penicillin, you can try other antibiotics such as tetracycline and streptomycin to avoid infection. Okay, try to avoid multiple sexual partners at the same time. Okay, that's a major problem. And also rapidly eradicating gonococci from infected individuals by means of early diagnosis and treatment. And you want to ask them a questionnaire to find the cases and also identify their contacts to find the reservoir, to eradicate the reservoir, so that finally the whole population will have fewer and fewer infection. Okay, so that's the last type of gram-negative cocci.